So welcome back everyone. So we are discussing a principle of inheritance and variation. In last class we have discussed basic things of Mendel. We have seen the failure of Mendel's work, success of Mendel's works, and the reason for selection of pea plant, and we have seen 7 contrasting character. Then we have seen some important terms. Then we have discussed Mendel's law of inheritance. First law we have discussed, right? Law of dominance we have discussed. So in this video lecture we will be talking about the next law. So we will continue with the Mendel's law of inheritance. So we have discussed Mendel's law of dominance, right? What happened in Mendel's law of dominance? When two contrasting character they come together in an organism, only one is expressed, and so visible effect, right? And it is known as dominant. The other remains hidden. That is recessive. So we have seen that, right? So today we will discuss second law. So first law we have completed. So second law known as law of segregation, or this law it is also called as purity of gametes. So law of segregation, Mendel's law of segregation, or purity of gametes. So remember which law is universally applicable, which law is not universally applicable. Okay. So first law, that is law of dominance, it is not universally applicable. We will see where it is not applicable. Okay. There is an exception. So what this law says, law of segregation or purity of gametes. This law states that both the parental alleles of F1 separates and express phenotypically in F2 generation. Whatever the resultant we get in F1 generation, that is our first generation, when we make the first generation self, when we do selfing the first generation, when we cross the F1 with the same parents, that is selfing, in the F2, that means in the next generation, that we get different characters. So we need to find out the phenotypic ratio and genotypic ratio in that case, right? First, what you need to do, you write it down, this law. Law of segregation or purity of gametes. So when you write it, you write like this way. Mendel law of segregation states that, then you continue. Under apostrophe says. When the law of segregation states that both the parental alleles of F1 separates and are expressed phenotypically in F2 generation. Don't get confused with alleles and gene. Okay, what is gene? It is a functional unit of inheritance. Okay, a part of the DNA which transcribe. Don't get confused. What is gene? Gene is a part of DNA which is inherited for generation to generation. That's it. Alleles is alternate form of gene. Okay, that is the alternate form of gene. A gene is a long segment of DNA. Alleles is a gene only, but it is an alternate form. Okay. So what happened in this case? Both the parental alleles of F1 separates when we make selfing. Okay. So when the first cross is made in the F1, the result that we get, if we make that selfing, if we cross that F1 with the same parents, so whatever the result we get, that would be different, and we need to find out the phenotypic ratio and genotypic ratio. We will see. Remember, this law is universally applicable. Sometimes for 1 mark question asks which law is not universally applicable, which one is not universally applicable. So remember, this law is universally applicable. Okay. So below we take a cross example. For example, This question, if this question comes, it comes for 5 marks. So explain the Mendel law of inheritance. When the question comes for 5 marks, you have to explain 3 laws along with the example. So example, we take a tall plant, homozygous tall and homozygous recessive, dwarf, right? So dominant character we always represent with capital letter, we have seen. So we are taking the homozygous tall plant and the homozygous dwarf plant. So, and the dwarf, that is recessive, that is dominant, we always represent with capital letter. And the recessive character we represent with small letter. If we take the dominant character tall, we write capital T, and when we write the recessive character, we write with the same letter, but it should be small letter. Okay, it is dwarf, that means recessive. We do not write with small d. Okay, there is a rule. If you write with this letter capital T, then we will represent it with the same letter, but it should be small t. Now if you take the flower color, which is our dominant flower color, purple, right? Purple is our dominant color. If we take the purple color, we take capital P. Okay, that is the dominant. What is the recessive of flower color? Purple is the dominant, recessive is white. So when we write a recessive character, we do not write with white W. Okay, we represent with the same letter, but it should be small letter. So recessive we always represent with small letter, but with respect to the dominant letters. Okay, but it should be small letter. So small p, that is our recessive character, right? So here, don't get confused. So example, we take a tall plant, P plant, and dwarf P plant. So dominant tall, recessive dwarf. That is our parents, okay? That is our parents. Now gametes, tall plant will give its gametes and the dwarf plant will give its gamete. So they will give its gamete 1 capital T. Dwarf plant, that is recessive, they will also give their gamete small t. So in F1, F1 means the first generation, when we make a cross between capital T, that is tall and dwarf, the result we get is all dominant, right? Dominant. Capital T is the dominant. So what should I write here? Tall or dwarf? T is the dominant. According to Mendel law of dominance, the dominant character always represent, always express and show visible effect, right? So capital T is the dominant here. So all plant will be tall in F1 generation. So to find out the ratio of phenotypic and genotypic, what is phenotypic again? External appearance, right? Genotypic means internal appearance, genetic makeup, genotype. So to find out the ratio, we need to make a self. We have to cross F1 generation with the same parent. Okay, so we say selfing of F1. So selfing of F1 means we will cross the F1 with the same parents. We cross the tall plant against the tall plant. So F2 result we represent in the form of Punnett square. So how we have to draw Punnett square? Just for us to understand how many boxes we have to draw. To understand that, just remember how many alleles are there here, 1 and 2. Okay, just multiply it with 2, same number. So 2 into 2, 4. There will be a 4 Punnett square. If the allele is 4 here, just multiply with the same number. Okay, if allele is 4, multiply with the same number. 4 into 4, 16. So there will be a 16 Punnett square. Okay, that is the technique to understand. So when we make a cross F1 with the same parent, we will get the ratio of phenotypic as well as genotypic. So we represent that in a Punnett square. So selfing F1, the F2 we get, I'm writing that side, you continue. So we have to draw 4 boxes, 4 rows and 4 columns. Okay, 1, 2, 3 and 4. Male characters will write at top part and the female character will write the below. Okay, so first we have this character. So put it here, capital T is there and small t is here. Okay, and the next female we have this character, capital T and small t. So when we make a cross, remember this first letter we have to cross this with all these two. Once we complete these two with this, we represent in this box. Now once we complete this, we again cross this small t with these two character. Okay, look at this. So first we make a cross, capital T, capital T. So write it down, capital T, capital T. So finish, then we make this, we make cross with this character. So capital T, small t. Okay, so this work is finished. Capital T is finished. It's done. Now come to the small t. So again we make cross with small t, capital T, small t and small t. So small t we can write this way also, or we can write that way also. Okay, it's the same thing. So let me write a capital T first. Okay, but we can write like that way also, but I will represent first capital letter. Okay, same thing. So then this cross is done. Now make this cross, small t, small t. Okay, so what we did a selfing here. We make a cross with the same parent. First parents we put in this box, capital T and small t. Second parents we put in this box, capital T, small t. Male, the first one is the male, second one is the female. Okay, so we make a cross. Now first, if find it out phenotypic ratio. Phenotypic ratio means external appearance. Okay, external appearance. So find it out how many tall and how many dwarf are there. How many tall and how many dwarf are there? So first find it out dominant character. Here we have dominant, that will be a tall. Again here it is a dominant, capital T is there, dominant, so that one will be a tall, right? Here again it is dominant, so it will be tall. So dominant character always show visible effect according to Mendel law of dominant, right? So capital T, capital T, capital T. So is there any dominant character here? No, there is no any dominant character. How we can understand this? By seeing the letters, whether it is in the capital letter or small letter, right? So these two, small t, are recessive. So that one will be our dwarf. So how many tall and how many dwarf? Okay. So we are discussing phenotype ratio. To find the phenotype ratio, find it out how many tall and how many dwarf are there. According to Mendel law of dominance, what it says, the dominant character always show effect, always show its visible effect, always represent its character. The recessive remains hidden. So how many talls are there? There we find a tall, capital T, that means the dominant. So dominant will always represent character. Again here it is tall and there is no any dominant character, there is no any capital letter. So how many tall? 1, 2 and 3. So 3 tall we find. How many small letter are there? How many recessive are there? 1, only 1. So that is phenotypic ratio, 3:1, 3 tall, 1 dwarf. When you write your answer, always below you represent tall in a bracket and 1 dwarf. Alright. If you still get confused here, find it out this character. So tall, capital T tall. The result F1 we get tall. 1 capital T, 1 small t. Remember the capital T, that means dominant. And the dominant character always show its visible effect. Okay, so there is again same capital T small t, there would be tall then dwarf. That is phenotype. Next you find it out genotype ratio. When you practice this, everything will be clear to you. Very easy. So to find it out the genotypic ratio, that is the genetic makeup, the internal structure, just find it out the same character, same letter. Just find it out the same letter, same allele. That means these two are alleles. Okay, together is a gene. These two are alleles. Don't get confused. Alleles are gene. They are gene only. They are part of gene only. These two are alleles. Okay, to find out the genotypic ratio, just find it out the same letter. So how many capital T and capital T are there? Find it out. Allele with the same letter. So there is only one capital T and capital T, right? There is only one. Below you don't have to write anything. Next we find it out how many capital T and small t are there. Here we have one. Again we have another one. So we have two with the same allele. That means with the same letter. Capital T small t, capital T small t. That one is small t, so we do not conclude here. So capital T small t, capital T small t. So we have two. Done. This is done, this is done, this is done. So we have only one small t, small t. So 1:2, 2:1. That is your genotypic ratio. To find it out genotypic ratio, just find it out the allele with the same letter. Just find it out the allele with the same letter. Okay, so this is our law of segregation or purity of gametes, and this law is universally applicable. So what happened? Why we are doing this? Why we have to find out the ratio? Why we have to do selfing? Why we have to find it out the F2 generation? Because our sentence says the F1, the result, whatever the resultant we get in F1, that is in first generation, when we make a cross, the F1 separates and express different characters. Okay, so why we are not doing Punnett square, why we are not doing selfing in case of Mendel law of dominance? Just read it out the sentence, what it says. In Mendel law of dominance, what it says, when two parents are crossed, that means when two alleles come together, only one is expressed, and that will be a dominant. The other which do not express and remains hidden, that one will be called as a recessive, and that is not universally applicable. It is not applicable to every experiment. Okay, there are some exceptions. Why we need to do this F2? Because according to our definition, according to our laws given by Mendel, it says that both alleles of F1 separates and express phenotypically in F2 generation. So F1 separates when we do selfing. That means when we cross F1 with the same parents. Okay, we are crossing this with the same parents. That means we are crossing with tall and tall, same parents. So when we cross F1, the F2, that is the next generation, we get this phenotype 3:1, genotype 1:2, 2:1. Okay, so remember this ratio. And that ratio is monohybrid ratio. Remember, monohybrid means the cross between same parent. Okay, the cross between same parent, monohybrid. So remember, that is monohybrid ratio. Next, when we discuss the next law, that will be a dihybrid, cross between two different character, two different parents. So that is our monohybrid ratio. You need to remember this ratio. Okay, easy. The phenotype is 3:1, genotype is 1:2:1. How we find it out? This way. Alright.